All right. I'm just going to back up in this PowerPoint to kind of set the stage for tonight's continuance of what we were supposed to do on Tuesday. If you really did watch that recording from the Monday-Wednesday class, and I hope you did, you had the first beginning part of my sympathetic anatomy and physiology review. And in that recording, you heard me remind you all that the autonomic nervous system is broken down into two divisions. And there is the parasympathetic division and the sympathetic division. The parasympathetic division and the sympathetic division are active in your body all the time. They are both active right now. It somewhat annoys me when an anatomy professor or physiology professor asks a question like, tell me, when is the sympathetic nervous system, quote, on? And students rattle off fight or flight or freeze or sex response, which means ejaculation. I don't like that question because the sympathetic nervous system is on, active in you right now. The better question to ask is, which division has more control over your physiology right now? I hope none of you feel terrified for your life right now. You shouldn't feel terrified. So right now, I hope you recognize that your parasympathetic division has more control over your physiology right now. It is your calm blue ocean division. It is what you use the most when you are resting, when you are eating food and digesting it. If you're running for your life from a zombie apocalypse, that's not an appropriate time to say, wait, time out, I need to eat something. No, there is no rest for the weary when you're running for your life. It is also the division you require in order to relax sphincters that control your urethra and your anus. So to go to the bathroom, to defecate, to urinate, you need the parasympathetic division. Reading. When we read, we have the luxury of having something up close in our vision to read it. When you are running for your life, you don't want up close vision. You want distance vision so you can see where your exit route is. You're running for your life. You need to find safety. When you read up close, you're not worried about an exit route, a safety route, an evacuation route. And your eyes adjust for that. When you are calm and resting and reading, you have pupillary constriction, and you have a smooth muscle inside your eye that controls the shape of your lens. This is called the ciliary muscle. And when you are doing up close reading, the ciliary muscle contracts. And when it contracts, it actually allows your lens to ball up. It becomes more rounded. And when your lens is more rounded, you are able to do up close reading. And this process is called accommodation. So that's why we have reading on here. Now the picture down at the bottom, you're seeing a 2-neuron chain. It sort of looks like dead stick figures, right? It looks like a head of a person and the feet, and then the feet of that person is communicating to the head of another dead stick figure. No, this is meant to represent a 2-neuron chain. Both divisions use a 2-neuron chain. And the anatomy review of tonight and earlier this week was to remind you where these cell bodies are found, where the neuronal cell bodies for both these neurons are found for both divisions. Knowing the anatomy is important. And it's basically, it comes down to, where do you find these neurons in your body? And let me give you a bigger picture of why this is important. If you are running for your life, what happens to your heart rate? When you're scared and terrified, we think of the beating heart. It feels like it's jumping out of your chest. Your heart rate goes up. The force of contraction goes up. Clearly, your sympathetic division is telling your heart to behave differently. It's telling your heart to behave like it's in a sympathetic moment. You're running for your life. You need more blood flow. You need a faster heart rate to ensure more blood flow to critical areas of your body. But when you're out of danger, we don't want our heart to keep beating like that. That's a lot of work on your heart. When it's time for us to rest and go to sleep, we don't want our heart to behave that way. We want it to reduce its beating rate and force of contraction. So my point is, we want these organs that are communicated to by these two divisions to know, to be able to know, right now I should behave this way. We're in a sympathetic moment. Now I should behave this way. We're in a parasympathetic moment. We need these organs that have both divisions communicating to it to know exactly which one has more control. And so even though they have some similarities, they have a 2-neuron chain, we appreciate that there needs to be some differences so that these organs know which division is communicating to them. So the first difference we're going to highlight, quickly review, is where you find the cell bodies. They're found in different places in your body. So they are housed. Their addresses are different in your body. That's good, right? We're separating them. The other thing you're going to see tonight is that the chemicals they use are different. The chemicals used to target these effectors, like your heart or blood vessels. And the proper name for these chemicals would be neurotransmitters. And if they use different neurotransmitters, they need to use different receptors. This is not a new concept for you. You know from Unit 1, hormones are chemical signals. And you know, in order for a target cell or gland to respond to a hormone, it needed a functional receptor. So these two divisions are found in different places in your body. They are going to use different chemicals, and they will use different receptors to respond to those chemicals. That's a good thing, because that means an organ like the heart that gets information from both knows exactly which division has more control at any given time. And the heart, if it could talk to you, would say, right then, I know how I should behave. I should behave in a parasympathetic fashion. My heart rate should go down. My force of contraction should just be strong enough to eject blood to the body. We don't need the pounding heart. Are there any questions before I move on? Okay, well then let me jump to what you should have learned from earlier this week. The video earlier this week showed you the 2-neuron chain, and to use the same colors, the first neuron is shown here in blue, and the second neuron is shown here in yellow. Instead of saying the first neuron of the 2-neuron chain for the sympathetic division, there's a better way for me to say that. I can say the sympathetic pre-ganglionic neuron. A ganglion is a collection of neuron cell bodies found outside of the peripheral nervous system. So the pre-ganglionic neuron is going to be the one that communicates to the second neuron. And the second neuron, its cell bodies are always found in the peripheral nervous system outside of the brain and spinal cord. For both divisions, the first neuron, its cell body, is always in the brain or spinal cord. The second neuron cell body is always found outside of the brain or spinal cord. So where do we find the sympathetic preganglionic cell body? It's in the spinal cord. Where exactly? All along the spinal cord? Nope. Only in levels T1 through L2. And at levels T1 through L2, you can see, when you took anatomy, you might have thought that the spinal cord looked like it was a butterfly shaped with the gray matter. But at levels T1 through L2, we see another butterfly wing, as I like to say. There's a third wing, a middle wing. And this is called the lateral gray horn, or cell column. Now, anatomists also discovered that there's another place in the spinal cord, more inferior, at levels S2, S3, S4. And physiologists said, oh, okay, there are these two areas where we get this lateral gray horn, but at levels T1 through L2, it is where we find our first neuron for the sympathetic division. At levels S2, S3, S4, which is the sacral lateral gray horn or column, it's for the parasympathetic division. So anatomists and physiologists said we should name these regions differently. The lateral gray horn at levels T1 through L2 is called properly the intermediolateral cell column, or IMLCC. It's a horrible name. I didn't make it up. That's the name. If at any time someone says, hey, Monica, I want to talk to you about the intermediolateral cell column, with your training, with you studying for this exam, you should be able to go, oh, IMLCC, I know where that is. It's in the spinal cord. It's at levels T1 through L2. And you're talking about the sympathetic nervous system, fight or flight. Great. What do you want to know? As soon as I say IMLCC, immediately, sympathetic division. That's what it means. That's what we're talking about. So I've drawn these blue dots for the cell bodies here in that lateral cell column. And the axons extend out of the spinal cord. Where do they go? They're going to terminate, synapse, communicate with the post-ganglionic cell bodies. All right, where do we find them? Where do they live in the body? What is their anatomy? What's their address? Okay, well, there are three general places where you find these post-ganglionic sympathetic neuronal cell bodies. They can be found in ganglia just next to your vertebrae. So it's not in the spinal cord, it's next to the vertebrae. And these ganglia are part of the sympathetic trunk. So we can find them in the sympathetic trunk ganglion. Ganglia is plural. And of course, this structure has another name called the paravertebral ganglia, which paravertebral means next to the vertebrae, next to your vertebral column. Another place where we find these post-ganglionic cell bodies is in the abdominal cavity, specifically on large blood vessels that come from the abdominal aorta. Large blood vessels that come off of the abdominal aorta include the celiac trunk, the superior mesenteric artery, the inferior mesenteric artery. And resting right on top of those big blood vessels is a cluster of neuron cell bodies. In the cat, if you were able to do cat dissections before COVID hit, these clusters of neuronal cell bodies are big enough to see with your naked eye. Here is a lifesaver, and you can see the hole in the lifesaver. Reduce that hole by half, and that's how big this cluster of neuronal cell bodies is in the cat. This hole represents the size of this cluster of cells in the human. That's big enough to see with your eye. You don't need a microscope. And these cells are sitting right on top of those blood vessels. And so the ganglion is named after the blood vessel it's sitting on. So you have the celiac ganglion, the superior mesenteric ganglion, and the inferior mesenteric ganglion. And that's why I've written those names here. Celiac, SM for superior mesenteric, IM for inferior mesenteric. The third place where we find the post-ganglionic cell bodies is the adrenal medulla. The students think, okay, Kara, the adrenal medulla is a gland, and these neurons are supposed to communicate to glands. So don't let this trick you. The adrenal medulla, the center of the adrenal gland, is actually the collection of neurons for the sympathetic division, and they're post-ganglionic. They are the post-ganglionic cells. And those cells are going to release their chemical signal, epinephrine, straight into the bloodstream, which means the epinephrine is no longer a neurotransmitter. It's instead now a hormone, because it was released into the bloodstream. So don't forget that one. The adrenal medulla tricks you. It makes you think that we don't have a 2-neuron chain. It makes you think, oh, it's just preganglionic communicating to the gland. You need to remember that the inside of that gland is actually a cluster of the second neuronal cell bodies. And that epinephrine that they release into the bloodstream travels throughout the body and can target other effectors in the body that are more distant. And that's what makes it a hormone that is being released. The last thing I want to tell you about on this slide, which comes from our earlier session in the week, are the involuntary effectors. What is it that the second neuron targets? Involuntary effectors. What is an effector? It's something in the body that will perform a function. What does something in your body? Skeletal muscle does something in your body. It makes you move. Smooth muscle does something for your body. It gives you gut motility. It can vasoconstrict blood vessels and change your blood pressure. We have glands in our body. They secrete substances. And we have a heart. Our heart does something for us. It beats our blood. And there's adipose tissue. You might be thinking, what does adipose tissue do for me? What function does it do for me? It just kind of sits there. It makes me fat. No, if you're running for your life, adipose tissue is an important fuel storage site. If you're running for your life and you don't have the luxury to stop and eat, you can burn fat as fuel. And mobilizing that fat is really important when you're running for your life. So adipose tissue does do something. So that means there are five effectors: skeletal muscle, smooth muscle, cardiac muscle, glands, and adipose tissue. Of those five, one is voluntary. You have conscious control over it. And that's skeletal muscle. That leaves us 4 involuntary effectors: smooth muscle, cardiac muscle, glands, adipose tissue. All 4 are targeted by your sympathetic division. All 4 are not targeted by the parasympathetic division. The parasympathetic division does not innervate, does not target adipose tissue. Thank goodness. Think about that. Thank goodness. Could you imagine going to sleep at night every night and waking up with 3 times more fat tissue than what you had when you went to sleep? Oh my goodness. Thank the heavens. Adipose tissue is not innervated by the parasympathetic division. The sympathetic division does innervate it. Oh, thank goodness. Because when I'm running for my life, I want to use those fat stores. I need that as fuel. So now I'm going to move on with my drawings. I'm going to draw with you all. Are there any questions, though, before I begin tonight's drawings? No. Okay. So the drawing I want to do with you all first is now going to address the chemistry that I referred to earlier. What are the chemical signals, the neurotransmitters, excuse me, that are used by the sympathetic division? All right, keeping with the same color scheme, our first neuron was drawn in blue. So again, not a dead stick figure. I'm drawing, the circle represents the neuronal cell body. The long line represents the axon. And then this portion down here is representing the terminal Bouton or synaptic terminus or synaptic knob. There are lots of names for that. I call it the terminal Bouton. That's the part of the neuron that releases neurotransmitter. Okay, so what neurotransmitter does the first neuron release? It releases acetylcholine. So down here, I'm going to keep my little legend of my pictures. So the blue dot represents acetylcholine. Okay. The word cholinergic is an adjective. This first neuron is a cholinergic neuron. Why am I calling it that? Because it releases acetylcholine. That's why it's a cholinergic neuron. That's the only neurotransmitter it releases. Neurons can't release more than one neurotransmitter type. So it's cholinergic for life. Okay, what happens when that first neuron releases acetylcholine on the second neuronal cell body? And let's say the second neuron is targeting the heart, okay? One of our involuntary effectors. We're going to try and get the heart rate up. We're in a sympathetic moment. Okay, we're not to the heart yet. Right now, we're focused on the first neuron communicating to the second one. The first neuron has released acetylcholine. All right, well, the second neuron, in order to respond to that, must need a functional receptor. We know that from Unit 1. So on the second neuron, it has receptors. And those receptors will bind the acetylcholine. So guess what? The second neuron has cholinergic receptors. What does that word cholinergic mean? It means it has receptors that can bind to acetylcholine. Okay, so let me add to my little legend down here. Acetylcholine receptor. All right, well, let me tell you a little bit about acetylcholine receptors. There are two kinds. There's one called nicotinic acetylcholine receptors and muscarinic acetylcholine receptors. Both bind acetylcholine. Their name tells you that. But their name also tells you that these receptors can bind a second chemical. Nicotinic acetylcholine receptors can bind nicotine as well. What will happen if the second neuron binds nicotine? It will behave just the same as if it bound acetylcholine itself. So nicotine is an agonist. That means it can bind to the same receptor and provoke the same response. That's not a new concept for you. No, it's not. Can you think of a parallel example from lecture unit 1 where you had some other molecule mimicking, pretending, if you will, as if it were the official molecule, and the downstream target didn't know any better because that mimicking molecule bound to the same receptor?
What's it? I know what it is. It's the stimulating.
Good. Keep going. You got it, Belinda. It's right there.
I know what it is.
Anybody want to help her out? She's so close.
But it's the TSI.
Good. Yes. Good.
You knew what it was, Belinda.
Yes, she did. She had it, huh? If it were a multiple choice question, she would have gotten it.
Exactly.
I know she would have. Okay, so yes, TSI was mimicking the actions of TSH. It wasn't TSH, but it was able to bind to the same receptor. And the poor thyroid gland didn't know any better. It doesn't have eyes. It can't look out and say, you TSI, you're trying to trick me, but I see. Thyroid gland doesn't have eyes. It only has a receptor, and if the receptor is bound, it should behave a certain way. So nicotine is an agonist. It can bind to nicotinic acetylcholine receptors, which means this second neuron, if it binds nicotine, it's going to say, okay, the first neuron told me to do something, I'm going to do it, and guess what? It's going to target the heart. This is a reason why people who smoke, they have a higher heart rate. It's because of the nicotine. They're not in a fight or flight. Is the irony lost on you? Why do people say they smoke? One of the biggest reasons why they say they smoke is because, quote, it calms their nerves. Is the irony lost on you? The very nicotine they're smoking stimulates this nerve, which in turn stimulates the heart, and their heart rate goes up. So their body is going to behave like it's in a sympathetic response, for crying out loud, when we're looking at the heart, of course. Silly people. It's less common now, but back in the day when I first started, more people in my class smoked. When we would have, I had to give breaks, not so much for people to go to the bathroom, but to go outside and smoke. It doesn't happen so much anymore because a lot of people don't smoke now, but also we're a smoke-free campus, so there's nowhere for you to smoke. But I used to tell those students when they'd be outside smoking, and I'd go outside to try and get some fresh air. That didn't happen. I'd say, you need to stop smoking. Have you learned nothing from my class? And they would joke and they would say, Kara, right now, smoking helps me maintain homeostasis. When I'm finished with physiology, then I'll stop smoking. I'll quit. So I thought that was funny. Now, muscarinic acetylcholine receptor, I will address that in a moment. Muscarinic acetylcholine receptors are not used by this division robustly. Listen to what I said. Muscarinic acetylcholine receptors are not used by the sympathetic division robustly. But I did not say that they weren't used at all. They are used, but in very special places in your body, and I will address that towards the end of tonight. So I'll come back to muscarinic acetylcholine receptors. Again, right now, where are we? We're communicating to the downstream neuron. The upstream neuron released acetylcholine, and the downstream neuron expresses nicotinic acetylcholine receptors. Now, in a lecture, maybe you've watched it already, called the Dendrites to Synapse lecture, you will hear, if you haven't watched it already, that the nicotinic acetylcholine receptor has a lot of subunits. To me, it sort of looks like a flower. And it has a portion of it that goes through the cell membrane. So it's an integral protein. So here's my phospholipid heads and the fatty acid tails. And when acetylcholine binds to this receptor, two acetylcholines bind to the receptor. And when it binds to the receptor, it turns out that this big protein that acts as a receptor is also a channel. They're the same entity. The receptor and the channel are the same entity. So when the acetylcholine binds, this channel opens and it allows sodium to diffuse into the cell, which is how sodium wants to move. It wants to go into the cell. This type of receptor, the way it works, receptor and channel, the same entity, is called ionotropic. Ionotropic. The receptor and the channel are the same entity. The word ionotropic tells you when the receptor binds the neurotransmitter, an ion channel is going to open. And in this case, the channel that opens allows sodium to come into the cell. And if sodium is coming into the cell, as asked earlier by a student in the class, this would lead the inside of the cell to become more positive, closer to threshold. This will generate an EPSP. And that means this second neuron is more likely to fire an action potential down the axon. Are there any questions so far about the first neuron and how it targets the second one for the sympathetic division? I'm going to write sympathetic up here so we remember that's the division we're talking about tonight.
So is the picture you drew at the bottom of the nicotinic receptor, is that pretty much like just like a zoomed in picture of the membrane on the or on the second neuron on the top?
Yes, correct.
Okay.
Thank you, Connor. Yes. There's a reason why I'm not an artist and I'm a scientist. You never want to play Pictionary with me. I'm not good at it at all. Okay. Well, now I'm going to start another slide, and now we're going to draw, or I'm going to draw and explain what the second neuron does to the heart. And I'm not just going to tell you what it does to the heart, I'm going to tell you what it does to the other involuntary effectors that I told you about in the beginning of this lecture. So give me a moment to just set my picture up a little bit. Again, we're talking about the second neuron. It is going to have to release a neurotransmitter. And we have involuntary effectors that it's going to target. We know that it will target the heart. So I'm going to draw my heart. We know that it will target glands, right? I said glands in the body like sweat glands, digestive system glands. There are glands in a lot of places in your body. So how do I draw a gland? My little drawing for gland is this. Why do I draw it that way? Because most sweat glands are coiled. So this is me drawing a coil. I know it's corny, it's stupid, but hey, it works. That's my gland, okay? And it doesn't just have to apply to a sweat gland. It could be the pancreas, which is a gland. It could be salivary glands. So that's my picture for a gland. Next, we have smooth muscle. So smooth muscle is spindle-shaped, and it is mononucleated. So that's my picture for smooth muscle. Now, this is where it gets interesting because we find smooth muscle in different places in our body. And it turns out we need to address these different places. For example, we have smooth muscle in the iris of our eye. We have two different kinds of smooth muscle, in fact. One type of smooth muscle in the iris of your eye, the colored part of your eye, is radial. It means it radiates out like spokes on a bike, which is why I drew it this way. We also find smooth muscle in your GI tract. So here is my drawing of the stomach leading to the intestines and then your anus. So that's my sad drawing for the GI tract. Okay, I know you can do better. I cannot. There's also smooth muscle in your blood vessels. So maybe I should draw that in red just so you know it's for blood vessels, right? Let's draw it in red for blood vessels. And there's also smooth muscle in your lungs. So I'm running out of room. Here's my trachea. Here is my lung. And another lung. It's not the kidney, it's lungs. All right, so the smooth muscle in your lungs lines your air passageways, your breathing tubes, if you want to think of it that way. Well, there's one more involuntary effector that I need to draw, and that's adipose tissue. And an adipocyte, when you look at it under a microscope, it looks like there's nothing inside of it, even though there is something inside of it. It's just a lot of lipid, and so the light shines right through it. And because the cell is filled up with so much lipid, the nucleus gets pushed off to the side. So it kind of looks like it's a ring, like a diamond ring. Some students in my class say, no, Kara, that looks like a boob with a nipple. I'm like, no, it's a ring. It's a diamond ring. Okay. So what neurotransmitter does this cell release, this post-ganglionic cell? It releases chemicals that are catecholamines. So that's my little legend. Well, what's an example of a catecholamine? Well, dopamine, norepinephrine, or epinephrine. This is not the first time you've heard those words. You learned about them in Unit 1 when you learned about amine-derived hormones. And you saw a slide with all three of these coming from tyrosine. And off to the side, you saw that tyrosine also helped make thyroid hormone. Not the first time you've seen these. So this neuron is called, what do you think it's called? Well, if the first neuron was called cholinergic, you might say, well, this one will be a catecholaminergic neuron. Yep, you're right, but that's a little difficult to say. So some students would say, well, most of these catecholamines have the word epinephrine in them. Norepinephrine, epinephrine. And these chemical signals used by the sympathetic division, we think of an adrenaline rush, right? Everyone wants an adrenaline. The word epinephrine, in Europe, they use adrenaline. So chemically, adrenaline and epinephrine are the same. Norepinephrine is the same as noradrenaline. So this neuron is an adrenergic meaning it's releasing an adrenaline-like neurotransmitter. It's adrenergic. So guess what kinds of receptors these involuntary effectors have to have? They need an adrenergic receptor, of course. They can't use a cholinergic receptor because that binds acetylcholine. Catecholamines are not acetylcholine. So they need adrenergic receptors. So here is my legend. The yellow Y, adrenergic receptor. So the heart has an adrenergic receptor. Actually, not just one, it has a lot of them. But I'm just going to draw one for simplicity. Smooth muscle has adrenergic receptors, and adipose tissue has adrenergic receptors. Okay. Well, unfortunately, the sympathetic division is just horribly complicated when it comes to its receptors. For acetylcholine, if you look at the previous slide, you heard me say there are two different kinds of receptors for acetylcholine. Just two. How many different receptors are there for these catecholamines? A lot more than two. A lot more than what I'm about to write for you. There's alpha 1, there's alpha 2, there's beta 1, beta 2, and beta 3. There are 5 receptors I need you to know about, and trust me, there are more. But these 5, they're like the big 5. These are the big 5 every good clinician should know about. Okay, Kara, fine. There are 5 adrenergic receptors on these involuntary effectors. So all 5 are on all of these, right? Nope. No. For these involuntary effectors, they might prefer one of these receptors and express it, have it on the surface the most. And we need to learn that. So let's start with the heart. I'm going to start with some easy questions. How many hearts do you have in your body? I'll wait.
One.
Good.
One.
Thank you, Alex. Alex is like, I know this one. One heart. What does it do for you? Any guesses?
Pumps blood through your body.
Good. It pumps blood through your body. Absolutely. In order for it to pump blood through your body, we understand it's beating, right? That's what we say when it's contracting. It's beating. So I want you to think of you have one beating heart. One beating heart. So it turns out the heart uses beta 1. Think 1 for heart, 1 heart, and B for beating. So when this heart binds a catecholamine, either dopamine or norepinephrine or epinephrine, any one of those 3, your heart rate and force of contraction goes up. Another word for heart rate is chronotropy. And another word for force of contraction is inotropy. Don't confuse that with ionotropic, which was described on the previous slide. Inotropy refers to the force of contraction. So our pounding heart is what we think of when we're in a sympathetic moment. Okay, so right about now, I should tell you that dopamine, norepinephrine, and epinephrine, just to make things even more complicated, and I apologize, but this is important. This is clinically important. Why? Why is it important, Kara? Well, first of all, let me remind you all, you're the ones that want to be future healthcare providers. So you're the one that made a decision to be in this prereq class. What I'm about to tell you, you need to know. It is clinically important because every day we are prescribing or using medications that target these receptors in our patients. And maybe you're saying, Kara, I'm not going to be a nurse. I'm going to be a physical therapist. Great. You still need to know this. Why? Why do I need to know it? You're going to be helping a patient that might be on this medication. And I hate to tell you this, but before anyone starts working with another person, we give them stacks of paperwork to go through, and we ask about their medical history. We ask about what drugs or prescriptions they might be on. So yeah, you do need to know it. Even if you're just going to help them with their stretching and flexibility and muscle strength, you need to know it. So these catecholamines, they don't all target all 5 of these receptors. Of course not. That'd be too easy, right? And I tried to tell you sympathetic division is awful. Dopamine will target alpha 1 and beta 1. Those are the only 2 receptors that dopamine communicates to. Norepinephrine is a little bit better. It communicates to alpha 1, alpha 2, and beta 1. Epinephrine communicates to all 5. Epinephrine's king. That's good because epinephrine is released both as a neurotransmitter and as a hormone from the adrenal medulla. And this is the reason why when the sympathetic division is more in control of your body, why your whole body reacts at the same time to this very intensive moment. We say en masse. En masse. I'm going to write it down here. En masse. That means all together. The parasympathetic division does not have that capability. Why? Why not? The parasympathetic division does not have a gland as part of its system. It does not have the equivalent of the adrenal medulla. There is no parasympathetic gland that releases acetylcholine into your bloodstream like a hormone. And you wouldn't want that because didn't I show you on this slide that acetylcholine is used as the first neurotransmitter? How confusing would that be for the heart? Am I supposed to behave like I'm in a resting and digesting moment or sympathetic moment, running for my life? You see, the parasympathetic division, when you review that anatomy, the parasympathetic division releases acetylcholine from both neurons. You see, chemically, both divisions start out the same. They even use the same nicotinic acetylcholine receptor on the second neuron. Chemically, they start out identical. Oh my goodness, what a dangerous setup. Because Kara, I heard you say that the downstream target involuntary effector should know which way it's supposed to work, how its physiology is supposed to change. And yet you're telling me both divisions start out using the same chemicals and same receptors. Yep. But it's the second neuron that's different. The second neuron, sympathetic division, releases catecholamines. The second neuron in the parasympathetic division continues with acetylcholine. And thank goodness the parasympathetic division does not have a gland to release copious amounts of acetylcholine into the bloodstream, because then none of your involuntary effectors would know how to behave. Why? Any guesses?
The mixed signals.
But why, Belinda?
Because one's telling them to increase and one's telling them to decrease, like the heartbeat, but they don't know which one to follow.
Correct. So let me add to your answer.
I don't know.
If acetylcholine was, you're fine. If acetylcholine was released into the bloodstream, it would bind to any acetylcholine receptor anywhere in the body. That means even acetylcholine receptors on the sympathetic post-ganglionic neuron, which would stimulate it and tell it to release catecholamines onto these involuntary effectors. So these involuntary effectors would get catecholamines binding to their adrenergic receptors, but they would also be receiving acetylcholine binding to their acetylcholine receptors. That's why they'd be very confused. So the parasympathetic division does not have a gland that releases acetylcholine into the bloodstream as a hormone. That does not exist. Just the sympathetic division has that anatomy. Thank you, adrenal medulla. Well, let's go to your lungs, shall we? How many lungs do you have? Assuming COVID hasn't ruined them, how many lungs do you have?
Two.
Good, two. Yes. What do they do for you, Jasmine?
Help
you breathe.
Yes, so they help you ventilate. And ventilation means you're breathing. You're inhaling, you're exhaling. So I want you to think of B for breathing, and 2 because you have 2 lungs. Now your beta 2 receptor on the smooth muscle in your lungs, beta 2 causes the smooth muscle to relax. And when it relaxes, beta 2 is always your muscle relaxer. And when the smooth muscle relaxes in your air passageways, guess what? You can breathe easier. Yes, you can. You ventilate more. There's little resistance. Just ask any person in the audience that has asthma. What do they take? They take their albuterol, their inhaler. What does that drug do? It stimulates beta 2 receptors. It's an agonist. And then they can breathe better. So beta 2 is your muscle relaxer. It makes you have bronchial dilation. Okay, what about fat? How much fat do you have? Do you have 1 adipocyte? Do you have 2? Or do you have a lot? A lot. Yes, unfortunately. Yes, we have certainly more than 2. So I want you to think of, we've got even more than 3. And if you've got too many adipocytes, is anybody else watching my 1,000-Pound Sisters? Anybody else Tammy and Amy fans? No?
Yeah.
Or you're not admitting to it? Monday nights. 1,000-pound sisters. These are morbidly obese individuals. So think obese, right? And I think O looks like the ring that I've made for this adipocyte. And then I think obese, I think B is in there. And we have a lot of fat cells. So I know it's corny, but it's better than nothing. Beta 3s. Beta 3 receptors are on adipocytes. And when they are stimulated, they release fat so that you can use it as fuel. Hey, do you all want to be rich? Don't become a nurse. Instead, go to graduate school and get your PhD. And please develop a drug that will specifically target beta 3 receptors on fat cells so that we burn our fat away. You'd make bank. And when you're rich and famous because you've made that instant fat loss pill, that magic pill that everyone's been thinking of and trying to formulate, you better thank me because I gave you the idea. So you have more lipolysis. You're using the lipid for fuel. That makes sense. You're running for your life. You need more fuel while you're running. Okay, what about glands? What about glands like your salivary glands or your pancreas? Digestive glands. Let's start with digestive glands. Well, alpha-1 receptors are used. Now, I don't have anything catchy to help you remember that, except I think saliva glands, and I think there's a lot of A in that word, saliva. So I think alpha-1. Now, when you're afraid, when you say we're in your speech class and you have to get up and give a speech, don't you get a little nervous? You're getting a little afraid. That's your sympathetic response. What happens to the saliva in your mouth? You don't make as much. It becomes thick and scant because saliva is needed for digesting carbohydrates in your mouth. Well, if you're scared running for your life, you're not digesting anything. So the amount of saliva goes down. We're inhibiting these glands from releasing digestive enzymes. So on glands like digestive glands, we decrease secretion, right? We're running for our life. Now is not the appropriate time to be releasing digestive enzymes. Hold on, I'm just making sure my son is not pinging my phone. Okay, nope, it's not. Okay, so alpha-1. Now, where else do we find alpha-1? Turns out we find alpha-1 on those radial smooth fibers in the iris of your eye. And when they're stimulated, it causes your pupil to expand. And we think of your eyes, or we say you're wild-eyed, like the deer stuck in headlights, right? You get pupillary dilation. I'm not kicked off of Zoom. I told you that this sometimes happens with my recording program, and I'm getting us right back in. There we go. I'm back. So pupillary dilation. Now, just to put this on here, you have another set of smooth muscle cells that go in a sphincter or circular fashion. Those do not have alpha-1. Those are under control of the parasympathetic division. So they would cause pupillary constriction. If you want to review the chemistry for those sphincter smooth muscle cells, you need to watch the other video where I review the anatomy and chemistry, physiology of the parasympathetic division. Okay, what about smooth muscle in your GI tract? Again, we're going to see alpha-1 receptors. And we should not be having food go from your stomach to your small intestine and then fecal material going from the small intestine to the large intestine. You should not be having increased gut motility when you're running for your life. That is a very awkward moment when you turn to the serial killer who's chasing you with a machete and you say, "Time out, I need to go big potty right now. Can you give me a break?" So alpha-1, and this is going to close sphincters. All right, so one more, and this is smooth muscle on blood vessels. Now, it is time for us to take a break. And when you come back from the break, I will finish the receptors found on blood vessels, because again, that's a messy story. Of course it is. It's the sympathetic division. I will also explain to you when I'm talking about blood vessels, I will explain the alpha-2 receptor, which is the only one that I haven't addressed yet. And then for the last part of tonight, I will address the special scenario where the sympathetic division does not release catecholamines and instead uses acetylcholine. Ugh. Kara, doesn't that cross the lines of communication? I know, but not as much as you might fear. So go have a nice break, and I need to finish something with Jasmine. So how about we come back at around 7:40. So take a nice 20-minute break. And when you come back, I probably will only need 20 more minutes of your time. So I suspect we'll be finished tonight by around 8 o'clock. So go have a break. And Jasmine, I'm going to pause my recording. Now that we're after the break, I need to go over the last bit on smooth muscle in blood vessels and explain the alpha-2 receptor and also the special case scenario for the sympathetic division. All right, let's first start with the blood vessel. And here is drawn a blood vessel and the terminal Bouton, and let me erase all of this because I already told you which receptors those catecholamines bind to. So let me make more room. What you're seeing in yellow is the post-ganglionic sympathetic neuron, the terminal Bouton, right? So let me show you here. The terminal Bouton, we're just more up close, and that terminal Bouton is releasing a catecholamine onto the smooth muscle found in this blood vessel. And you're seeing that this blood vessel has alpha-1 receptors. In fact, blood vessels have both alpha-1 and beta-2. I'll address that in just a moment. But in this picture, this simplistic picture, you're seeing epinephrine binding to the alpha-1 receptor. And when alpha-1 receptors are bound on smooth muscle, it causes contraction. So this would lead to vasoconstriction. That's why I wrote vasoconstrict on the right side of the blood vessel. Now in red, I am drawing the alpha-1 receptor on the blood vessel smooth muscle found in the tunica media. But in orange, there is a different receptor that I drew. In orange, I drew the alpha-2 receptor. So let me use my little legend again. And you're seeing that epinephrine binds to alpha 2, which is found on the terminal Bouton of this post-ganglionic cell. This receptor is participating in negative feedback inhibition. This is not a new concept for you. You know, if we use the example of HPT pathway, you know that the thyroid hormone feeds back to the hypothalamus and tells the hypothalamus to reduce TRH levels. How does the hypothalamus know this? Because its receptors are populated by thyroid hormone. And you heard me say in that lecture, this ensures that we don't get too much of the hormone released. So the epinephrine in this picture is binding both to alpha-1 receptors on the smooth muscle in the tunica media of this blood vessel, but it is also binding to alpha-2 receptors. And this is making sure that this neuron does not keep dumping too much epinephrine into the synaptic cleft and keep dumping and keep dumping. If alpha-2 receptors are bound, it reduces the amount of neurotransmitter released by this neuron. Why would that be helpful in this scenario? If a blood vessel stays vasoconstricted too much for too long, the downstream tissue that would be served by that blood vessel is not getting adequate blood flow, is not getting nutrients, is not getting oxygen, and that tissue will die. So when we are in a sympathetic moment, blood flow to unnecessary tissues is reduced. What is unnecessary when you're running for your life? Blood flow to your ovaries. Right now, you shouldn't even be entertaining ovulating if you're running for your life. Blood flow to your gonads in general. I'm just using that as an example. At that moment in time, those are not necessary organs. So they should not be getting a large amount of blood flow. Instead, we should be redirecting blood flow to more critical organs like the heart, like skeletal muscle, because you're running for your life. Now, the thing is, once you're out of this dangerous situation, of course you want blood flow to go back to those other parts of your body. That's true. We hope they're still alive, though, because if you've been in this very stressful moment, this life-threatening moment for a long time, those tissues might have been starving for oxygen and nutrients at this point, and they might have started to die, and death is irreversible. And if you're saying to me, Kara, we are not in stressful situations that long in our everyday life. We are like the gazelles on the African prairie. The lioness chases us, we evade the lioness, and we live. So sympathetic moment over, it maybe lasts some minutes, but you're making it sound like the sympathetic moment can last for hours or days. It can, and it does. We are one of the few species on this planet that has chronic stress issues. We are not an animal that only has a sympathetic moment because we're being preyed upon. We have chronic stress, and our sympathetic division does not know the difference between I was almost in a car accident versus I gotta pay the rent, I have kids that I need to get to the doctor, I might lose my job because everything's shut down with COVID. Your sympathetic division doesn't know the difference. It's you perceiving a threat and your body is responding. So this alpha 2 helps, as if to say, if it could talk, it would say, hey, you're right, you should redirect blood to other more critical areas of the body. However, we don't need to shut down blood flow to those other parts of the body entirely. They still need to get something, because once you survive this mess, those other organs are going to be useful, and we would like them to not be dead. This will not be the only time you hear me tell you this. When we get to Unit 4, you're going to learn that when you're in a fight or flight response, turns out your kidneys aren't thought of as being critical. I know. Your body would say, they're just filtering the blood and making urine. When you're running for your life, we don't need to make urine right now. In fact, urine reflects losing blood volume. And when you're running for your life, you want your blood pressure up. You want blood volume up. So making urine is a bad thing. And yet your kidneys are going to say, yeah, but when you survive this, you're going to need us. You don't want us to be in renal failure when you're done having your sympathetic moment. And you're going to see in Unit 4 that the kidneys have their own protective mechanism. They don't have this alpha 2, but they have other mechanisms to ensure, okay, you might be reducing how much blood we get. Fine, we understand we're not critical right now. But eventually when you survive, you're going to need us again. Because for your daily life, we are critical. Are there any questions about alpha 2 participating in this negative feedback inhibition to regulate the amount of catecholamine, targeting, most notably, the smooth muscle and blood vessels? Okay, well then let me tell you another part of this blood vessel story. Turns out blood vessels can have alpha 1, but also beta 2 on their tunica media. So they don't have as many beta 2. They have a lot more alpha 1. A lot more. And you'll remember from one of my previous slides that alpha 1 and beta 2 bind to norepinephrine. Back up. You'll remember that alpha 1 and beta 2 bind to epinephrine. Both of them bind to epinephrine. So if both of them bind epinephrine, like I've shown in this picture, then that means how will the blood vessel, smooth muscle, know how to react? If alpha 1 causes constriction and beta 2 causes dilation, how will this smooth muscle in this blood vessel know to constrict or vasodilate? And the answer is this. Beta 2 has higher affinity. So there are fewer beta 2s, but they bind to epinephrine first. That's what we mean by higher affinity. So when epinephrine levels are kind of low, beta 2 receptors bind epinephrine first, and this blood vessel would vasodilate. If epinephrine levels are high, then all of these receptors would be bound, and now numbers win. There are more alpha 1 receptors than beta 2. So then if all of these receptors are bound, the alpha 2s would cause vasoconstriction. And then the students say, well, which blood vessel are we talking about? Doesn't matter. You should know that blood vessels can have different numbers of these. All I want you to know when it comes to this scenario, I just want you to know we can have both. We frequently do have both. Beta 2 are fewer in number, but they bind more readily to epinephrine. If epinephrine levels are low, we get vasodilation. And you can think of, well, when would epinephrine levels be low? Think of when you're exercising. You're not necessarily afraid for your life, yet exercising is a mild sympathetic response. Think of low levels of epinephrine, and clearly when you're exercising, blood vessels being vasodilated would be a good thing. Not just to your skeletal muscle, but also to your skin, because it helps with thermoregulation while you're exercising. Okay. When epinephrine levels are high, a severe sympathetic response, all those receptors are now bound, and now numbers win. More alpha 1 means we see alpha 1 response, which is vasoconstriction. Any questions about that?
Oh, ma'am.
Oh, ma'am.
Okay. Well, then the last issue I need to address before I let you go tonight is your skin. So it turns out in your skin, you have involuntary effectors in your skin. You have sweat glands. You have blood vessels in your skin with smooth muscle, cutaneous blood vessels, and you have another smooth muscle that makes your hair stand on end. Now, when you're in a sympathetic response, you get goosebumps. Other animals that have a lot of fur, their hair stands on end. We're not very furry animals, but wouldn't it be kind of fun if your hair stood on end when you got scared, just out like this? Wouldn't that be fun? Just for one day even. So that's your erector pili, smooth muscle. And when you are having a raging sympathetic moment, ever notice, for example, when people are in shock, people go into shock when they are in a life-threatening situation, for example. Sympathetic division kicks in high gear. And have you ever touched someone who's in shock? They feel cold. Why do they feel cold? These blood vessels going to the skin at that raging sympathetic moment, alpha 1 receptors are really responding, going to vasoconstrict. Or I should say, let me back up. Blood vessels leading to these blood vessels would be vasoconstricted. They're cold and clammy, they're producing sweat. So why is this a special case scenario? Back when you were an embryo, back when you were a developing human being, the neurons that grew to innervate these involuntary effectors actually grew from parasympathetic centers. So the chemistry is parasympathetic. That's where it came from. So in this picture, you're seeing this post-ganglionic cell releasing acetylcholine. And these involuntary effectors out here in your skin have muscarinic acetylcholine receptors, one of the other types of acetylcholine receptors I told you about earlier. What is muscarine? It is another chemical that can act as an agonist. Where do you get muscarine? Mushrooms. The Amanita muscarina mushroom. I'm sure you don't know what that looks like, but I'm sure you know better than to walk randomly outside, pick up mushrooms from the ground, and eat them. I'm sure you know some mushrooms are poisonous. And the Amanita muscarina mushroom is poisonous. Why? It makes muscarine. And muscarine can bind to your muscarinic acetylcholine receptors, wherever they are in the body. And the parasympathetic division uses acetylcholine and muscarinic acetylcholine receptors abundantly. That's its chemistry. So on your heart, you have muscarinic acetylcholine receptors. If you eat muscarine, your heart is now being told, behave in a parasympathetic moment. Your heart rate goes down. Your GI tract is being told to behave in a parasympathetic moment, which means digesting. You're going to have increased gut motility and release of digestive enzymes. Anywhere you have muscarinic acetylcholine receptors, it will bind muscarine, and you will die a very messy death. You will ooze from every pore and orifice. Tear glands, salivary glands, digestive glands, yes, even the sweat glands in your skin. Your skin involuntary effectors, when you were first developing, were innervated by parasympathetic systems. The chemistry is parasympathetic. But somewhere in your development, in utero, different brain centers said, you know what? We're going to take over. Thank you, parasympathetic division, for growing all the neurons and your chemistry. You did all the hard work wiring everything in the skin, but we're going to be in charge. Think about how different we would look if these skin involuntary effectors were still under control of your parasympathetic division. You would be producing sweat right now. You would be having vasoconstriction of these blood vessels right now, so you would feel cold and clammy to the touch, even when you were in a calm blue ocean state. And your hair would be sticking on end. You'd have goosebumps all the time. When you were scared, you wouldn't have goosebumps. That would be funny, wouldn't it? So the chemistry is all parasympathetic for these involuntary effectors, but they are under the command of the sympathetic division. I know that's ugly, I know that's messy, but that is how you developed. And that's all I have for tonight. Do you have